becoming really clear early in the conversation here is that cancer is us. I think a lot of times when people think about cancer, the layperson, they want to fight cancer, destroy cancer. What you're making clear throughout the first 15, 20 minutes here is the fact that these are our own cells.
Yeah.
That are in the first part when you're describing, they're going back to a more primitive form. And then you describe with the metastasis, the fact these macrophages are starting to behave, well, they're converting over to cancer cells. So it's our own body that is changing.
Right.
So when we talk about fighting cancer, essentially we're talking about fighting our own body.
And as long as you keep that body full of sugar and glutamine, the sugar and the high fructose foods that we eat, I mean, you're just letting that thing go crazy. So that's clear. What's happened now? Why did our ancestors and some of the primitive Aboriginal tribes have such a low level of cancer? Because of their environment. If you're in an environment where highly processed carbohydrate foods are in minimal supply, and you have a lot of exercise, we have a surveillance system in our body. And if we have a cell where the mitochondria may slowly become corrupted, that cell is consumed for the good of the whole. Our macrophages come in, see that cell, it's not performing, it's not living up to the society requirements, it's eaten. Excuse me. And then the molecules from that cell that was eaten are now distributed throughout the body for the rest of the cell. So you say, well, why did cancer become so prevalent over the last 50 or 60? I mean, we've had cancer since Neolithic times, but it was very rare. It was extremely rare, and now it's overtaking heart disease as the number one killer in Western societies. So what's going on? And we're allowing our cells to become lazy and fat, and we're not surveilling the body the way we should. It's hard to ferret out weak cells if there's always a surfeit of carbohydrate glucose in the bloodstream, because the rest of the cells become they're less proficient in weeding out the weak, the lame, the unfit, when everybody is fat and happy with all the sugar in our system. So there's no need to ferret out these dysregulated cells and then all of a sudden you find yourself with it spread all over your body, or you really have a big problem. So you can go back and look at the way most cancer is very rare in organisms that live in their natural environments. All right, wolves rarely had cancer. Very few of our primate relatives, the chimps, the gorillas, the orangutans, cancer is very rare in these animal species. Just like our Aboriginal past, cancer is extremely rare. It's only within the last, say, 50 or 60 years, especially within the last 30 or 40 years, that we see this. Why is this? What is going on? It's not genetic. This whole nonsense about cancer being genetic is crazy. It's environmental. Wake up, people. It's an environmental situation. As soon as the Food and Drug Administration unleashed high fructose corn syrup, and as soon as our society became more dependent on technology, we lack our exercise. We're filling our bodies with glucose, and sugar is not a carcinogen, but it creates systemic inflammation. Systemic inflammation will damage oxidative phosphorylation. As systemic inflammation allows you to be infected by viruses, oncogenic viruses. Your bodies simply become weaker when you're in these kinds of states. As I said before, we have an obesity epidemic. This propels people for type 2 diabetes, dementia, cancer, cardiovascular disease. It's all of these diseases together are called chronic diseases from our diet and lifestyle. Okay, diet and lifestyle are responsible for this problem. And, now you try to figure out, what are we going to do to manage the system? Well, you could do prevention. As you know, most people don't care about prevention. Some do, most people don't. And then when you have cancer, people want some sort of a rapid cure, and you get all these toxic things that you're given when you do standard of care, and you have a bad problem, you make it worse. But you're right, Jesse. It's ourselves. It's our body. It's the way we live in our society that's contributing to a lot of this problem.
There's some really interesting research with cells, and this is where they took different organelles. Actually, it was the cytoplasm and the nucleus, and then they put different tumors into the cytoplasm and the nucleus. Talk about how that worked and what they found out from that.
Yeah, well, that's the nuclear transfer experiments. I summarized all of those, as I mentioned in the last. Maybe I did. I don't remember. But those nuclear transfer experiments were a backbreaker to this whole view that cancer is a genetic disease. These studies were done by Dr. Kinnell at the, he was down at Louisiana. He since went to the Minnesota. He passed away a couple of years ago. McKinnell's work on the frogs. And then there was Jim Morgan's group at St. Jude's, and there was Rudy Yanish's group down here at MIT, Beatrice Mintz's group, Israel and Schaefer's group from the University of Vermont, Shea and Wimberlin from University of Texas. I mean, all of these groups had done these kinds of experiments where they would take the nucleus of a tumor cell and put it into the cytoplasm. They took the nucleus of a nucleus of a tumor cell, put it into a normal cytoplasm, and you would get no dysregulated growth. On the other hand, if you took the normal nucleus and put it into the tumor cytoplasm, you got dysregulated growth. And I documented how these studies were done over and over again, repeated time and time again. Okay, all different types of cancers, all different types of protocols. And as I said, what were they doing? They were simply asking whether or not a tumor nucleus could direct normal development or not. And the answer was the tumor nucleus was suppressed in its abnormal growth. So the cytoplasm was controlling the cytoplasm was controlling the regulation of growth. And that's exactly what I said, because in the cytoplasm are the mitochondria, and the mitochondria control the cell cycle. And if the mitochondria are damaged, it doesn't make, if the nucleus is so you'd have all these nuclear gene mutations, and you put it in a normal cytoplasm, and you get normal regulated growth. I have a picture of it around here somewhere. I should have had it, but the picture says 1,000 words. You can follow the picture. I published it in this paper here. The folks can read all of the details of these experiments. If you go and read the detailed documentation of these with all the references, I even have it in my book. I have a whole chapter on the nuclear mitochondrial transfer experiments. Also, if you have a cell that's dysregulated in growth, and you say, well, we're going to take out clean out the bad mitochondria and put new mitochondria in, the cell becomes regulated in its growth. And then if I have a slow growing or a normal cell and I put abnormal mitochondria in there, it becomes dysregulated in its growth. So it's just everything is controlled by the mitochondria in the cytoplasm, not by the genes in the nucleus. So this is the key that tells us that cancer cannot be a nuclear-driven genetic mutation disease, and that is a mitochondrial metabolic disease, and that changes the whole playing field about how cancer should be treated, managed, and viewed. And once that comes around, the whole system will change to a more rational way to treat this without toxicity. So until that becomes more widely known, we're going to suffer through what we're currently having.
And where this can become confusing for people is the fact that with the new theory you're proposing, there still is genetic changes as cancer develops. The difference is, with the conventional wisdom and theory, they're saying the cause of cancer is genetic. In your case, you're just saying that genetic changes happen as cancer develops.
Yeah. I mean, they come down. So the cancer cells are throwing out what we call reactive oxygen species. So if the mitochondria are abnormal, oxygen comes in, and instead of serving as an acceptor for electrons to generate ATP, they form these ROS, reactive oxygen species, which are like metabolic bombs. They destroy the proteins, they destroy the DNA, they cause mutations in the DNA, they cause abnormalities in the lipids, and they kind of just disrupt everything, right? So the mitochondria become damaged. They produce ROS. ROS are carcinogenic and mutagenic. What does that mean? It means that the abnormal metabolites, these ROS coming out of the mitochondria, are causing the mutations as downstream effects in the nucleus, as downstream. So yes, when you look under big gene sequencings like they do down here at the Broad Center or wherever these big gene sequencing, they find thousands and millions of genetic defects in the nucleus of tumor cells. And precision medicine says we need to focus on those unique kinds of mutations to try to manage cancer. That's crazy. They're all downstream stuff. And you can see why we have 1,600 people a day. They're not dying from cancer because these therapies, precision medicine, all this kind of stuff you hear about, it's not working because it's based on a flawed theory. And in this paper here, Dr. Christos Xenopoulos, world leader in mitochondrial energy metabolism, and I compared and contrast the view of cancer as a genetic disease or cancer as a mitochondrial metabolic disease. And it becomes clear to everyone within a reasonable level of education and functional brain cells that cancer cannot be a genetic disease, period. Okay? Period. It's not a genetic disease. Yet, people would say, how is it possible you can say that when I go down to all the top cancer centers and they're all telling me that it's because they don't read the scientific literature. Either they don't read it or they don't understand it or they don't want to know about it. So what am I supposed to do about that? I mean, I'm telling you what's going on. People can listen. They can make up their own minds. They can read the papers themselves and come to their own conclusion.
The beautiful thing about your theory, and we're going to get to treatment here, the beautiful thing about that as well is the fact that this is very non-toxic and it applies to cancers across the board.
Yeah. And I think that's really important, Jesse, because, and that's another thing that is holding us back massively. That you think brain cancer is different from breast cancer, different from lung cancer, different from colon cancer, different from bladder cancer, different from melanoma, different from leukemias, different from all these. I have a leukemia, breast cancer. They're all very, very similar. I showed all that evidence. We went back and looked at every one of the major cancers, and they all have defects in the number, structure, and function of their mitochondria. That means every one of these major cancers is dependent on glucose and glutamine, right? I mean, this is scary. It's very scary. You're telling me that all these cancers, unite the tribes. Bring all these breast cancer, bring all the colon, the brain, bring them all together in one table and sit around and saying, what do we all have in common, folks? Okay, you think your brain cancer is different from my breast cancer, from my colon cancer? They're all fermenters. They all have damaged respiration and they're all fermenting. So are you telling me a similar kind of therapy could be effective in managing all these kinds of cancers? And the answer is absolutely. How do you know? Because I've done it. I've tested it in the lab. We can't find any cancer that can live without glucose and glutamine. So what does that say? I mean, it's not like, oh, they're so sophisticated. They're so tough. Oh, they're so wily. That's all bullshit. They're just not reading the literature on what you should know to know about the biology of the cell. Are there any therapies anywhere in any major clinic on the planet that is reducing glucose and glutamine while transitioning the body over to ketones, because ketones cannot be fermented? And the answer is no. It's not being done anywhere. Are there clinical trials thinking about this? Yes. What does that mean? Well, we're going to use standard of care and then maybe throw in some sort of a diet. That's going to have a minor effect. You got to do what I say. You have to do the way we're working, and we're developing the global therapy for cancer because we understand the biology of the problem. We know how to tackle this. Will it be successful on every human being that would come in that would have cancer? Probably not. But I can tell you it's going to do a hell of a lot better than everything that we have currently as a standard of care. And I know this because I've tested. I've been spending many, many years looking at these kinds of things, and we're shocked by the fact that it cannot be adapted in clinics. And why you figure it out? Why you ask the people what's going on here? Why is this not being done?